Hello, good morning. I am Rogelio Flores from Universidad Autonoma Potosi, and I'm going to present our project titled Molecular Printing Polymers for Local and Controlled Release of Antibiotics, a new strategy against antimicrobial-resistant bacteria. Today, I would like to highlight the importance of Diabetes mellitus as of the most significant non-communicable disease worldwide. According to International Health Organization, hundreds of millions of people are currently living with diabetes, and this number continues to rise. Diabetes is characterized by elevated blood glucose levels resulting from impaired insulin production, insulin resistant, or both. Over time, uncontrolled diabetes can lead to several complications, including cardiovascular disease, kidney failure, blindness, neuropathy, or lower limb amputation. These complications significantly reduce quality of life and increase healthcare costs worldwide. In Mexico, it's not it's the same, it's the same scenario. More than 21 million people live with diabetes, making the second lead cause of death. One of the complications, the most severe and costly complication of type 2 diabetes mellitus definitely is diabetes foot ulcer. This disease, its development results from a complex interaction among peripheral vascular disease, neuropathy, immune dysfunction, and infection. Once a wound develops, bacterial colonization and infection further impair of healing process, often leading to chronic ulcer, hospitalization, and even amputation. In this context, our work explored a novel approach for the local and controlled delivery of antibiotics as a promising strategy to improve diabetic wound management and combat antimicrobial resistance. At present, the management of diabetic foot ulcer remains a major clinical challenge. According to Senatec in Mexico, treatments begin with a comprehensive etiology assessment, including neurological, vascular, and biomechanical evaluation. When infection is suspected, clinical examination and microbiological testing are essential to guide therapy. Current management relies heavily on systematic antibiotics and surgical debridement. However, the systematic administration may result in limited drug concentration at the wound site and contribute antimicrobial resistance. This limitation highlights the need for innovation strategies capable of delivering antibiotics directly to the gut in a controlled and sustained manner. Definitely, local drug delivery systems offer an attractive alternative by delivering the therapeutic agent directly to the site of action. This approach improves drug concentration at the gut, enhances therapeutic effectiveness, minimizes system side effects, and reduces the need for repeated administration. One of the type of system of delivery antibiotics. It's the molecular imprinted polymers or MIPs. MIPs are fabricated using a template molecular, functional monomer, cross-linker, porogen, and initiator. During polymerization, the template interacts with the functional monomer to form a pre-polymerization complex. After polymer formation, the template is removed, leaving height-specific recognition cavities that match the target molecular in size, shape, and chemical function. This unique combination of specificity, stability, and control release makes MIPS particularly attractive for the treatment of infected diabetes goods. Before presenting our current work, I would like to highlight a previous study conducted by our research group on the development of molecular imprinted polymer for control release ciprofloxacin. The most important results is the material exhibits significant antimicrobial activity against Escherichia coli and Staphylococcus aureus, while maintains a low cytotoxicity toward dermal fibroblasts, demonstrate both efficacy and biocompatibility. This previous work represents a key milestone to starting a point for research present today. The purpose of this investigation is to synthesize and characterize molecular imprinted polymers for local and control release of amoxicillin and piperacillin tazobactam, and to evaluate their efficacy against multi-drug resistant bacteria. To develop our control relief system, we synthesize MIPs and the corresponding non-imprinted polymers, NIPs, use the co-precipitation method, like this slide indicates. To rational design of MIP, we first evaluate the potential interaction between the template moleculars and select functional monomer. It's a lactic acid. As shown this slide, both amoxicillin and piperacillin tazobactam possess multiple function groups capable of forming non-covalent interactions, primarily hydrogen bonds with lactic acid. This interaction is essential because they guide the formation of polymerization complex and ultimately determine the specificity of recognition sites with polymer matrix. We validate the analytical method by HPLC with DID, and this is results. We successful synthesize MIPs and NIPs using co-precipitation method to template to monomer ratio of 1 to 4 was selected to promote formation stable interactions between antibiotic moleculars and polymer network. The absorption study demonstrates an excellent affinity between the MIP and their target antibiotics. Absorption efficiency range to 87 to 98%, indicate that the polymer were highly effective in capturing and retaining the template molecules. The highest absorption was observed with piperacillin, reached 97%, while other formulation exceed absorb about 87%. 100%. The results confirm this successful generation of a specific recognition sites and highlight the potential of these materials has a high capacity drug delivery system. To confirm the success formation of MIP, we performed a 8-year FTIR analysis and compare the spectra of the free template moleculars and the imprinted polymer before template removal and the polymers after extraction. Showing this spectra, characteristic absorption bands associated with amoxicillin and piperacillin tazobactam are clearly observed in imprint polymer prior to the plate removal. These bands correspond mainly to hydroxyl, carbonyl, amide, and aromatic functional groups involved in the interaction between the template moleculars and the polymer matrix. And these results provide a strong evidence successful molecular imprinting. These slides show the same characterization of the synthesized MIPs and NIPs. This micrograph reveals that all materials consist glomerulous and quasi-spherical particles with raw and heterogeneous surface, which are desirable characteristics for absorption and drug loading application. Using potentiometric titration, we determined the pH PPC of the MIP was 3.9, corresponding to intersection of points of the titration curve. This parameter is particularly important because defines the pH at which the polymer surface has a net zero charge. This means pH values about 3.9, such a physiological condition evaluated in this study. The polymer surface becomes increasingly negative charged due to the protonation of carboxylic groups present in the polymer matrix. This behavior is directly influence the interaction between the polymer and antibiotic molecule. With respect the table, as shown in the table, amoxicillin, piperacillin, and tazobactam exhibits different ionization states depends of pKa values and the surrender pH. At the pH 5.4 and especially 7.4, piperacillin and tazobactam predominantly contains negatively charged carboxylated groups, while amoxicillin may exist in protonized or neutral forms. As shown in the kinetic profiles, all polymers exhibit a very rapid absorption phase during the first minutes of contact, following a gradual approach equilibrium. The absorption capacity establishes at approximately 5.5 to 6.2 milligrams per gram, with equilibrium being reached with about 50 minutes. The release studies were performed under physiological condition with this model. condition at 37 degrees Celsius and 150 RPM using phosphate buffer saline and pH 5.4 and 7.4. This experiment is to understand the release mechanisms, including zero order, first order, Iguchi, and Cosmeyer-Peppas model. The release profile reveals the clear pH-dependent behavior. In general, higher cumulative drug release was observed at pH 5.4 compared with 7.4, which is particularly revealed because infected diabetics both often exhibit an acid microenvironment. As a result, the Cosmeyer-Peppas models was used to determine the release mechanism through release exponent and provide insight into the fuse and polymer relation process government drug delivered from this MIPS-based system. In this experiment, the object was determined with MIP could enhance antimicrobial performance of the loading antibiotics compared their corresponding NIP. And we used two reference strains were selected Staphylococcus aureus and Escherichia coli. And the main of results is the MIPS results demonstrate that the synthesized MIPS retain a very good biological activity, antimicrobial, and this is for the antibiotics may provide an effective platform for control antimicrobial delivery. In this section, we evaluate the cytotoxicity using normal dermal fibroblast and the MTT assay. And the main results, all polymers exhibit cell viability values about 100%, indicate that these materials not caused cytotoxic effects. According to international standards, the materials are considered non-cytotoxic when cell viability remains about 7%. And in our study, our values were substantially higher than this threshold and demonstrating excellent cytocompatibility. In conclusion, the MIPS and NIPS show no evidence of cytotoxic toward dermal fibroblasts. In conclusion, the absorption process antibiotics onto the MIPS was found to primarily governed by chemoabsorption mechanisms occurring between the polymer recognition sites. Release profiles were obtained of MIPS and revealed significant difference depending on the combination of antibiotics incorporated to single polymer matrix. In addition, pH was shown to play the key role modulation behavior, and the MIPS are good antimicrobial efficiency and very good, indicate excellent biocompatibility with cells. Thank you so much for